Hi, my name is Nur Ali Atika binti Nazar Azman from UiTM Kuala Pilah, and in this video we'll talk about the molecular identification of Cryptosporidium parvum from 3 recreational areas in Negeri Sembilan. The water recreational area are popular among local visitors and tourists for leisure and weekend recreational area. However, many do not know that recreational area have many waterborne parasites such as Cryptosporidium and Cardia, which came from the droppings of animal and human excretion. So thus, increase of human activity in the recreational area will contribute the presence of waterborne parasites. So the first objective of this research is to detect the Cryptosporidium parvum from 3 recreational areas in Negeri Sembilan using staining and microscopic technique. Second is to identify Cryptosporidium parvum from the 3 recreational areas in Negeri Sembilan using nested PCR. So basically the methodology is first is water sample collection, filtration of water sample, and then detect the oocysts by modified Zeal nasal staining, and then we identify the Cryptosporidium under microscope and proceed to DNA extraction for further and nested PCR, and then observe under gel electrophoresis. So nested PCR is basically 2 sets of primer. So this is the primer name specific for Cryptosporidium parvum. In table 2, it shows that nested PCR component for 15 microliter, and the PCR condition for primary and secondary, we can see the annealing temperature is 55 degrees Celsius for both primary and secondary cycle. So from the study, we get that the morphological detection of Cryptosporidium parvum oocysts in water sample from the 3 recreational areas in Negeri Sembilan has been done by microscopy, as it most widely used for the diagnosis. So it's due to relative low in cost. So under 1,000 magnification, Cryptosporidium were found and by the stain population rat with Zeal nasal stain. So they had a spherical form and were between 4 to 6 micrometer in size. So based on the gel analysis, the band of Cryptosporidium parvum came out as expected band, which are 655 base pair respectively. But there was a band at the negative control as shown in figure 1, and negative control indicate the contamination of workflow. Other than that, there was also formation of primer dimer on the gel. So this occur because low of aseptic technique, as nested PCR has highly chances of contamination, which can occur during the transfer of the first product into the second tube for the second round of implication. Meanwhile, for primer dimer appear because the reduces of the final yield of the target DNA product. So the primers anneal to each other rather than attached to the target in the template DNA. Troubleshoot was performed, and some initiative of troubleshoot which changed the nucleus-free water and diluting the primers again with the new nucleus-free water. Meanwhile, for primer dimer, we optimizing the primer annealing temperature in PCR, which can increase the yield and specificity of the amplified DNA product. In figure 2, this was after troubleshooting. There is no band at the negative control, and at lane 345 there was a band of expected base pair. So in conclusion, this study confirms that presence of Cryptosporidium parvum from the 3 recreational areas in Negeri Sembilan by staining and microscopic technique, which can pose potential threat to human health through contamination water sources. So molecular identification by nested PCR has also shown the confirmatory result, where the Cryptosporidium parvum presents from the 3 water recreational areas in Negeri Sembilan with the appear of expected band. Thus, early detection of Cryptosporidium parvum oocysts is crucial for developing preventive measures to monitor recreational area as well as human health and environment. The result of this study can be used to further exploration of Cryptosporidium parvum from all water recreational area in Negeri Sembilan, and other alternative is using PCR restriction fragment length polymorphism RFLP analysis of Cryptosporidium outer wall protein genes is as it more simple and rapid.